This is some of the evidence that I spoke about in the previous segment. This is a picture taken from one of the Mars Exploration Program missions, Mars Pathfinder, and it's a picture of twin peaks, and there's the two peaks right there. And you can see Mars really doesn't look that different than many parts of Arizona. But clearly there's no civilization here. There's no cities, there's no roads, there's nothing that indicates that there's anything alive. And essentially the entire planet doesn't look exactly like this, but there's no civilization anywhere that we've seen. So the question, though, is not, is there civilization on Mars? That's not our question anymore. The question is there life? There doesn't have to be a civilization in order for there to be life. But again, if we look here, we're not seeing giraffes, or we're not seeing hippopotamuses or something like this. We're not going to see anything like that, we expect, because we haven't seen anything like that at all when we were taking pictures of the entire planet. But if there are no creatures that we see and can understand, then does that mean there can't be life? Well, obviously not. You know that many living things are too small to see. And so that's the kind of creature that we would be looking for on Mars. That's what we're expecting to find if there's anything that's alive on the planet. And those creatures, therefore, would be defined technically as microorganisms or microbes. And a microbe is simply any organism, which is again a creature made out of one or more cells, that's too small to be seen with the naked eye. Some microbes are made out of one cell, most I guess are, but some are multicellular. There are some creatures that you can't see, some little worms. Nematode worms are so small that you can't actually see them, but they're more than one cell. So the question then is this: what is a cell? Now, you've heard that term before, and I'm sure even in elementary school you put together pictures of cells or projects where you showed what the things were inside cells and so on. But we need to go through that again, because we have to come at this from the professional aspect. And so a lot of what I'm going to be talking about are things that you have seen before, but please pay attention, because there are things that I may be telling you that you may not have heard before. Now, you've probably, and I'm expecting you already to know, that there are two major types of cells on the planet. Cells like this, represented by this picture, and cells like this, represented by these two pictures. Now, the one on the left, if we look at this creature on the left, this picture here that I'm circling with the cursor is a single-celled organism. This particular picture, by the way, is a micrograph. This is a picture from a transmission electron microscope. And you look in here and it looks quite different than this. This is a picture from a light microscope. And you see inside here there's all these bubbles and things and objects and so forth. This thing's really a cool thing. It squeezes shut and then opens and squeezes shut and opens and so on. There's nothing like that in this particular thing. So here's the thing. This cell is a fundamentally different type of creature than this cell. Now, what I would like you to do is to go to your book, not the internet. I want you to go to your book and look up all the differences between these two types of cells. This cell that's depicted on the left are called prokaryotic. This cell depicted on the right are called eukaryotic. Now, you probably already know a number of things about the primary differences between those two things. What I would like you to do is go to your book and find something that you don't know about the difference between them. One of the reasons I'm asking you to do it this way is because almost all of the resources that you're going to find from a Google search are going to get it wrong. Almost all. Because here's what they say. The primary difference between this prokaryote and this eukaryote is that the prokaryotes do not have a nucleus and the eukaryotes do. That is not the primary difference. So what I'd like you to do is go to your text, list all the differences you can find, especially find at least one that you did not know before you took this class. Now, a second question I'm going to ask. Look at these two pictures here and here, just the micrographs, okay? The electron one and the light one. Are these cells the same size? If they're not the same size, which one is smaller? And how much smaller is it? All the evidence that you need is in this picture. Everything you need is on this slide. Do not look it up. Don't go to your book. Don't go to the internet. Don't go anywhere. I want you to, from this slide, and the evidence that's on this slide, determine which of these is bigger or smaller, or are they the same size? And if one is bigger than the other, how much bigger is it? Okay, so not a bad idea at this point maybe to stop and do that before we move on. Do that, get that written down, and then we'll continue on. Okay, at this stage, you should have written all of the answers to those two questions out, listing all the differences between prokaryotes and eukaryotes that you can find in the textbook, and also determining which of those two cells was bigger and how much bigger was it if one of them was bigger. So if you haven't done that, stop and do it, because what I want to do now is to move on, assuming that you've got that information. And here is the quintessential picture of a eukaryotic cell. And again, there are high school textbooks and other resources on the internet that say the difference between prokaryotes and eukaryotes is that the prokaryotes don't have a nucleus and the eukaryotes do. Here's the nucleus, this big purple thing in this picture. This little object down here is called a nucleolus. That's where the ribosomes are made. And you might recognize this right here, and there's another one here and another one here. Those are all mitochondria, the powerhouse of the cell. That's one that most people know. And then we have the rough endoplasmic reticulum and the smooth endoplasmic reticulum out here. And then you've got the Golgi apparatus. You got various vesicles and all kinds of other things that this eukaryotic cell has that the prokaryotic cell lacks. So if you discovered in your research there that one of the key differences between prokaryotic and eukaryotic cells is that eukaryotic cells have these kind of things that are called organelles, there's a word, then that's also a correct difference between prokaryote and eukaryote. Eukaryotes have organelles, not just a nucleus, and prokaryotes don't. But neither of those, to a modern professional biologist, is the definitional difference between the two. The key difference, which I hope you found, is that the prokaryotic cells lack this cytoskeleton. That's the key difference. Eukaryotic cells have a cytoskeleton. What a cytoskeleton is a network of protein fibrils that run throughout the entire cell and give it its shape and change its shape and hold the organelles where they are, move the organelles, move other things around within that cell. Prokaryotes don't have those at all. And that is the key difference. That's what you should have found. You should have found that the key difference between a prokaryote and eukaryote is that eukaryotes have cytoskeleton. Why is it that that's the key difference? Well, the whole reason why it is that a eukaryote has a nucleus and a prokaryote doesn't is because of the cytoskeleton. The whole reason why a eukaryote has organelles and a prokaryote doesn't is because of the cytoskeleton. It's the cytoskeleton that actually constructs all of this stuff. And that's why the prokaryotes don't have it, because it doesn't have the object that constructs it all. So that's the thing. A cytoskeleton is this network of protein fibrils within the cell, and the organelles are identifiable structures that are membrane-bound. That's the technical definition. They have to be membrane-bound, which means some things that we call organelles, like ribosomes, are not technically organelles, but I'm not going to worry too much about that. But at any rate, this is the picture I want you to have in your mind of a eukaryotic cell, at least for now. I'm going to change that later. And the key thing, when somebody asks you what is the difference between prokaryote and eukaryote, your answer is cytoskeleton, not nucleus, and not organelle. That's the professional definition. Okay, so what should we be looking for on Mars? Should we be looking for eukaryotic cells or prokaryotic cells? We don't know. But the eukaryotic cells are much more complex than the prokaryotic cells. And the principle of simplicity suggests that maybe we should be looking for prokaryote-like creatures first before we would expect to see any eukaryotic things. Because one would expect the prokaryotes to evolve before the eukaryotes because they're so much less complex. So what kinds of creatures do we have on the planet that could, for example, be prokaryotic cells? Well, here are some examples. This row are pictures of various types that are of a particular type of bacteria. And the bacteria is Leptospira. And it causes a disease in humans called leptospirosis. This picture right here is of a type of microscope that we call a scanning electron microscope. Here's another picture from a scanning electron microscope. And here's a picture from a transmission electron microscope. The difference is this. In these two pictures here, we're bombarding the surface of these microbes with electrons. The electrons scatter off of them, sort of like a mirror, they bounce off, and we're collecting those electrons as they bounce off. Here, we're shooting electrons through, so it's more like an X-ray. We shoot electrons through the microbe, and we can see the details from that. So this is like looking at it from the surface, and this is like looking at it with an X-ray. Okay, now notice the scale bar. You see here? This is 1 micrometer. That's a lowercase Greek letter mu, means micro. I want you to look up exactly what micro means, okay? Because we're going to see that. That's something, again, you've had chemistry. That's the assumption. You've had chemistry. that you should know what microbe means. If you don't know what micro means, then I want you to look that up, and I want you to compare it to nano, pico, and milli. Okay, so those are the things I want you to do, because we're going to use this a lot. But notice, pardon me, all of these pictures, they have these scale bars. That's what you should have used to determine what the scale was, or what the size of those cells were in the previous slide. Okay, so these are Leptospira. These organisms here, you notice, are not these long, skinny spiral things. They're these rod-shaped creatures, and they cause food poisoning. They're called Salmonella. And then these down here are two different things that are not actually living by the definition of most biologists. This is vaccinia virus, which causes a disease called smallpox, and this is long, skinny thing, notice though much smaller than Leptospira, is the Ebola virus. So now I would like you to take a look at these pictures and compare them to the pictures I just showed you of Leptospira and Salmonella in particular. You'll notice they're pretty similar, right? They're these blobs that look like they're just sort of sitting on top of the surface of this rock, and these are obviously scanning electron micrographs. Here's another one that looks a bit like Salmonella, kind of a rod-shaped thing. Now, notice the scale bar, though. This is 200 nanometers. Is that more or less than a micrometer? Now, if you look those things up, as I was asking, you'll realize this is much, much smaller than a micron, 1 micrometer. This is 200 nanometers. This is 2/10 of a micrometer. So these are much smaller than what we were seeing in the previous picture. But what's interesting about them is this. They come from an object that's called Allen Hills 84001. It's called that because it was found in the Allen Hills region of Antarctica in 1984, and it was the first of its type found in that spot at that date. The reason it's interesting is it's a meteorite. There's evidence it's a meteorite. The outside you can actually see here has been burned, and there's other evidence that it actually came through the atmosphere. I'm not going to go through that. And the interior has been sampled. What's interesting about the interior is this. The interior rock, when it forms, it'll form around and form little pockets of atmosphere and the thing that it in the place where it formed. And so that's one of the ways that we know what the early Earth's atmosphere was like. What's interesting about this rock is there's never been a time in the Earth's atmosphere that it resembles the atmosphere that was trapped inside this rock. But the atmosphere trapped inside this rock is identical to the atmosphere on Mars. And it's different than every other atmosphere of every other planetary body or moon that's in this solar system. So there's strong evidence, among other things, not just that, that this actually came from Mars. It got knocked off of that planet and got into a ballistic orbit around the sun and landed on Earth in the Allen Hills region of Antarctica and was found in 1984. How long it had been there? Millions of years. There's evidence for that and so forth. If you want to see that evidence, I actually have this paper up in the website, and you can actually download it and read it if you want. But I want to focus not on whether or not this thing came from Mars, because essentially the evidence is quite clear that it did. I want to focus on what are these things? And where were they deposited? That's what's interesting. So on this rock right here, on this meteorite, we have things that look a lot like these. A lot like these. And in fact, they're not too much different in size than these things, which are viruses and not bacteria. But still, they look like microbes. And there's other evidence, which I'm not going to go through, but there's other evidence that suggests that these objects, whatever they are, were deposited when the rock was on Mars. I want to just give you a sense of some of the evidence for that. They're shocked and ripped in a certain way that occurred when the rock itself exploded off of Mars. So they must have been on the surface of the planet before the rock was jettisoned into space. So in this case then, it's been suggested by some in NASA that these things are Martian microbes that were essentially knocked all the way to Earth. Now, they weren't alive when they got knocked to Earth, but these are fossils. That's what they're suggesting. These are fossil microbes. Now, the evidence here is stronger than just the fact that these look like microbes that we have on Earth. There's other things. There's chemical tests that they did. These things have a certain type of carbon compound that is suggestive of life after it's been decayed and so forth. So there's other things as well. But that's what they suggested. Okay, now, Whether or not you believe that is not the point. Whether or not scientists believe that is not the point. What I want to get at is, what are we talking about here? How are we speaking? Because that's going to help us understand what's going on. Here's what we're not doing. We're not forming an opinion, a strong opinion, until we've analyzed the evidence. That's a very different way of thinking than a lot of people are trained to think or practice thinking. We hold our opinion back, and that's difficult to do. We hold our opinion back until we can assess the evidence and look at it from all different perspectives and use the baloney detection kit on it. For example, one of the things the baloney detection kit suggests is that we should consider multiple hypotheses. Right? So we're going to think about that here in just a moment. But here's a question. Let's talk about language. Language is important. When I call something a hypothesis or something a theory or something a fact or a law, I got to know what I'm talking about, and you've got to know what I'm talking about. Otherwise, we can't communicate. So what I want to ask is this. NASA scientists suggest that these are fossilized Martian microbes. That statement, NASA scientists suggest that these are fossilized Martian microbes. What kind of statement is it? What is that? Is it a statement of theory? Is it a statement of a law? Is it a statement of a hypothesis, a principle, or a fact? Now, if you ask different people, you're going to get different answers. I'm not interested in the variety of people's answers. What I want to know is, how is it that we as professional scientists use those terms? I'm not saying that other people are wrong. I'm not saying that they shouldn't use those terms in the way that they use those terms. But I am saying, if we're going to communicate between ourselves and with them, we've got to know how it is that we are defining those terms. Now, I can tell you from experience that the vast majority of politicians, journalists, and I think the vast majority of people that I've spoken to would say that that statement is a theory. Most scientists would not. A scientist generally does not call that a theory. NASA scientists suggest that these are Martian microbes. That's not, to a scientist, a theory, although it would be to, for example, a journalist. I'm not saying the journalist is wrong, just saying they're using that term differently. To a scientist, that would be a hypothesis. We would be stating it as a question, really almost as it could be, not that we think it is, which makes it exactly not a theory. I'll explain that in a moment. But the point is this. We've made the suggestion, now what do we do? Well, again, you can form an opinion right off the bat if you want, but that's dangerous, we've discovered. If you're in a position, for example, where you're working in an ER as the ER attending physician, and somebody gets brought in and they're unconscious, but they otherwise don't look bad, there's been no trauma, they got no broken bones, they're not bleeding anywhere, but you can't figure out why they're unconscious. If you start guessing, almost certainly, we know from experience, that person's going to die. You have to know that you have to get evidence, and you have to know what evidence you need in order to determine what exactly is going on with this person. That's the way that we think, and that's the way that we can find the correct answers in certain circumstances. It doesn't always work, but it's the way that we approach things in science. So this is the question then. Let's talk about what a hypothesis is, so we know when we use that term, we know what we're talking about. Now, your book defines hypothesis as, quote, a testable statement that explains a phenomena or set of observations. Okay? So that then is, by your textbook's definition, of hypothesis. I find this definition to be reasonably correct in the following sense. When I speak to my colleagues in the profession, we generally tend to use the word hypothesis in that way. Okay? Now here's the thing. When somebody makes a testable statement, is this testable? These are Martian microbes. Yes, it's testable in the sense that if they weren't Martian microbes, we should be able to show that they weren't using evidence. So that's what we mean by testability. So here then is, if you wish, one of the central tenets of scientific thought. Whenever anyone states a statement that we see as a hypothesis in our sense, then it must be tested. And it must be tested using evidence from the real world. And it has to be discarded. It's dangerous not to discard a hypothesis if it is shown to be logically inconsistent or it fails to fit the evidence from the real world. That is the central tenet. Now, how you feel about that personally, I don't know, but I can tell you this is how we think in the profession. Whenever somebody makes a statement that's a hypothesis, we use these two criteria to test it. First, is it logical? Is it logically consistent? Now, sometimes that can get tricky because sometimes your intuition you mistake for logic. And that happened, for example, when we started studying quantum mechanics. And early on, people were considering, oh, quantum mechanics or even relativity didn't make sense. It was illogical because one of the crazy things, for example, relativity said was that the faster you go, the heavier, the more massive you become. Well, it turned out that that wasn't logic, that was intuition. Our intuition was wrong. Our logic was expressed by the mathematics, and the mathematics was perfect. So that's the point. We look at things from a logical standpoint, not from an intuitive standpoint. And don't mistake intuition for logic. Now, how then do we do this? How do we determine what's logical? Well, again, that's typically mathematical. But then there's this other issue of evidence from the real world. It has to fit evidence. We talk about what evidence is. Evidence is something that information that comes directly from nature herself. If somebody says these are Martian microbes, then we would make predictions as to, okay, if they really were Martian microbes, then these other things should be visible. And we should be able to see these things. And we should not see other things. And if we see the things that contradict that those are Martian microbes, we have to discard the idea that they're Martian microbes. That's the way scientists think. Now, this is tricky, though, because the point is, it sounds like we're saying you have to believe this and you have to not believe this. That's exactly not what we're saying. You are free to believe whatever you wish at all times. But as a professional, your job is to assess the logical consistency of something and the evidence for something. That's your job as a professional. You yourself can hold whatever opinion you wish, but if you're going to be a professional scientist, this is how we actually assess our information. And that's the definition almost of what science is.